Good morning to all. Annai Arts and Science College takes the liberty to upgrade the students, research scholars, and faculty members of different streams from various institutions all over the India via the International Virtual Conference on Recent Advances in Biochemical and Material Sciences 2020. This conference aim is to bring together illustrious minds in broad areas of physical, chemical, and biological sciences. I welcome you all for the 2 days international virtual conference scheduled to be conducted by our institution's science departments on 24th and 25th of November 2020. Prayer don't need to be long and eloquent. They only need to come from a sincere and humble heart. Let's start the day with a prayer song by a college choir.
Sing the wondrous love of Jesus, sing His mercy and His grace. In the mansion bright and blessed, He will bring for us a place when we all get to heaven. What a day of rejoicing that will be when we all see Jesus, then sing and shout the victory. While we walk the pilgrim pathway, cloud will overspread the sky. But when traveling days are over, nor the shadow nor the skies, when we all get to heaven, what a day of rejoicing that will be when we all see Jesus. We'll sing and shout the victory.
Thank you, choir. There is a quote by the great scientist Albert Einstein: "Science can flourish only in an atmosphere of free speech." I would like to call our principal ma'am to deliver the welcome address.
Happy morning to one and all. I take this opportunity and feel privileged to honor and welcome you all to the 2-day international conference on recent advances in biomedical and material science 2020. On behalf of Annai Arts and Science College, I cordially welcome our chairman, Chevalier Dr. N.R. Dhanabalan, our dynamic secretary, Mr. N.R.D. Premkumar, joint secretary, Dr. P.E.R. Premchand, and today's speakers of the session 1, Ms. Devika Kishan, Research Associate, University of Connecticut, United States of America, and second session speaker, Dr. Prem Saran Thirumalai, Assistant Professor, Department of Botany, Dayalbagh Educational Institute, Uttar Pradesh, India. I also gladly welcome Vice Principal and IQAC Coordinator, all the heads of the various departments, faculty, delegates and participants from the various colleges, staff and students to this 2-day international conference on recent advances in biomedical and material sciences 2020. A warm welcome and thank you to each and every one of you for joining with us today. I am indeed very happy that Department of Chemistry, Physics and Biochemistry along with IQAC has successfully organizing this 2-day international conference. I welcome and thank the distinguished speakers from abroad who will share their knowledge and experience in educating the young minds. The objective of this conference is to promote, encourage, and bring together researchers, scientists, philanthropists, medical service providers to share their knowledge in recent advances made in their respective fields. In biomedical sciences, research is going on in various fields like biomaterials, drug delivery, stem cells, and cancer research, advances in clinical diagnosis, herbal medicine, and pharmaceutical science. During this COVID-19, we all are eagerly waiting for a vaccine to come and save the world. And material science is also a huge field. There are many areas of research, but currently the major focus and topics that are trending and are based on the energy system for future energy security. Some of the fields of research in material science is biomaterials and tissue engineering, ceramics and glasses, nanotechnology, functional materials, and engineering alloys. I sincerely wish that this two-day international conference will be a great success, not only has a chance to share knowledge, but also create a new devoted resources. I wish this two-day international conference a great success. Thank you.
Thank you, ma'am. Now, I would like to call Dr. L. Parimala, ma'am, Head Department of Chemistry, to deliver a short note about the departments and the International Virtual Conference 2020. Over to Parimala, ma'am.
Ma'am, unmute பண்ணி பேசுங்க ma'am.
Parimala ma'am, unmute and speak ma'am.
Happy morning to one and all present in this forum. Annai Arts and Science College is a reputed arts and science college affiliated to University of Madras, Chennai, founded in the year 1997 by National Ratham Educational Trust. The institution is striving hard to fulfill the vision of the founder, Chevalier Dr. N.R. Dhanapalan, education to all. Our college aims to impart value-based and comprehensive education to both men and women with the motto of seek, strive, and succeed. To keep the student community and educators engaged and updated during this pandemic, our college has been organizing webinars, workshops, and FDP through an online platform. One such initiative is the International Virtual Conference on Recent Advances in Biochemical and Material Sciences, organized by the Departments of Chemistry, Physics, and Biochemistry on 24th and 25th November 2020. The Department of Biochemistry was established in the year 1997, offering UG program, and it was elevated to a BG department in the year 2005. The Department of Chemistry and Physics were established in the year 2015 and 2017 respectively. Every year, our students have secured university ranks in Madras University examinations. The departments organize seminars, symposium, conference, and intercollegiate competitions every year through the department clubs. The main focus of the departments is to mold the students to be intellectually compatible, socially responsible, and professionally capable to face the challenges in the competitive world. The conference would help to receive intelligent insights from some of the leading professionals, which can be a stimulating factor for personal as well as professional growth for the students and young researchers. This conference aims to bring together illustrious minds in the broad areas of physical, chemical, and biological sciences. The speakers of the conference are Ms. Devika Krishnan, Research Associate, University of Connecticut, US. Second session speaker is Dr. Prem Saran Tirumalai, Assistant Professor, Department of Botany, Dayal Educational Institute, Uttar Pradesh, India. Third session speaker is Tyagarajan, Assistant Professor, Coordinator, Department of Microbiology, Immunology, Northern Border University, Kingdom of Saudi Arabia. Fourth session speaker is Dr. Pachamuthu Kandasamy, Senior Director, Head of Medicinal Chemistry, Wave Life Sciences, Cambridge, US. Session 5 speaker is Mr. Vel Murugan N. Ramachandra, Vice President, Engineering and R&D, AMP MedTech, USA. Session 6 speaker is Anandakumar Pandi, Assistant Professor, Department of Biochemistry, AIMS, Jharkhand, India. Session 7 speaker is Kanagadara, Dr. Kanagadara Ma'am, Associate Professor of Physics, Savita School of Engineering, Chennai. I welcome all the delegates and participants once again to this international conference. Thank you.
Thank you, ma'am. I would like to call Dr. R. Harini, ma'am, Assistant Professor, Department of Biochemistry, to introduce the guest speaker of the session 1, Ms. Devika Kishan, ma'am. Over to Harini, ma'am.
Good morning and a warm welcome to everyone. Myself, Dr. Harini, Assistant Professor, Department of Biochemistry, feeling delighted to be a part of this program. On behalf of the management, principal, IQAC, faculty members, and students, I take immense pleasure to extend a warm welcome to our resource person, Ms. Devika Krishnan. I would like to present an introduction about our distinguished resource person. At present, she is working as a research associate at University of Connecticut, US. She worked as junior engineer at Corindus Vascular Robotics. Currently, she is doing her PhD on biomolecular mechanics and nanotechnology at Arizona State University. She completed her master's degree at Boston University. As a life science researcher, she is much passionate about investigating creative solutions for medical product development. With this brief introduction, I am thanking you, ma'am, for generously accepting to be a keynote speaker in this webinar session on nanotechnology in biomedical research. Now, I request Ms. Devika Krishnan to take over the session. Ma'am, please.
Thank you so much for the welcome. I will start with sharing my screen. Okay, so my name is Devika. I am doing research at the University of Connecticut in biomedical engineering. And today I'm going to talk about nanotechnology in biomedical research. Now, this is quite a comprehensive field and a very emerging field, but I will try to do justice to it in today's presentation. We can, of course, not go into all the details, but we will touch upon the different particles which form part of nanotechnology, and then we'll go into some applications, and then I will end with a few specific applications which will give you a flavor of how nanotechnology actually is used in biomedical research. Okay, so first of all is to understand, let's understand the scale of things, right? What do we mean by nanotechnology? The definition of nanotechnology is basically the study and the dimension below 100 nanometers. So we are studying particles which are below 100 nanometers. So if you just look at the slide here, you can see the ant is about 5 millimeters. So we are there still at the micros level. Then you go down to red blood cells, which are found in our body, and it's about 7 to 8 micrometers. There we are going down into the micrometer range. And then we go below, we come into the nanometer range, and that's where you have your DNA, which is our genetic material. That's about 2.5 nanometers in diameter. So you can just imagine a magnitude of scale that we are going down below to study these nanoparticles. And I would like you to think about the tools that are actually required to study these particles. So it's not just that nanotechnology has been accompanied with these nanoparticles. Yes, it started with the nanoparticles. So there is a brief history is that Richard Feynman talked about nanoparticles in 1959, but the term nanotechnology only came in much, much later. And some disturbance. Okay, sorry about that. But so basically nanotechnology was coined much after it was first talked about. So it first started with the nanomaterials itself, but then with the instrumentation, the tools that were allowed us to visualize this, to look at these particles, that's what really brought nanotechnology from just the particles, use of the particles itself, to being used in biomedical research or in any sort of world application itself. An interesting fact is that, so stained glass, if you look at stained glass, how people created stained glass in like 16th, 17th century is by adding gold and silver particles. So when they add gold to glass, they saw a particular color. When they added silver, they saw a particular color. And it was much later that people realized that the gold and silver acts as quantum dots. But that was the principle. So it was still being used in the 16th and 17th century, just that people didn't understand why or what it was. And the reason is visualization. That particular addition allowed us to see something, and that is something I'd like you to think about as we go through this presentation. Okay, so this slide was taken from actually a marketing paper, but I liked that they kind of visualize the different scales. So let's move on to the main hallmark of nanotechnology, which is basically the nanoparticles themselves, right? So that's what forms that was the beginning, the first part of nanotechnology. And they can be broadly classified into organic nanoparticles, inorganic nanoparticles, and carbon-based nanoparticles. I can't say that all nanotechnology specialists follow this classification, but for our purpose, we will just classify it into these three parts. So organic nanoparticles are mostly what is ideally required or would be required for biological systems, because they are closest to a biological system. So they're basically made from biology like biocompatible molecules. So some things like liposomes or micelles and things like that. So we'll go a little bit into detail in the next slide, but even DNA. So there is a field called that DNA nanotechnology, which basically uses synthetic DNA to self-assemble and form nanoparticles. So these have the advantage that they are closer to the biological system. So there will be less attack from the biological system on these particles. Then you have the inorganic nanoparticles, which was the first few nanoparticles, the metallic and the metal oxide nanoparticles, the derivatives of that, which are still being used, as we will see, but for different purposes. And then you have the carbon-based nanoparticles, which is like carbon nanotubes or graphene molecules, which we have probably studied about in school, if you remember the fullerene particles, but now at a much, much smaller scale. Okay, so this image gives us an idea about these different visual cue to these different nanoparticles. So this here is the liposomes. The first figure here is the liposome. So basically what it is, our body is made up of cells. Cells have these membranes, and these membranes are made of these molecules called lipid molecules, which have a hydrophobic head, they have a hydrophilic head and a hydrophobic tail, and they assemble. If you give them particular protocol, they assemble into these structures, these spheres, where the hydrophobic ends are together and the hydrophilic ends are together. And these structures can act as carriers. So you can put drugs in them, and they can be used to deliver drugs or to target certain cells. So that's one of the applications of liposomes. Okay, and then now this is a carbon nanotube. This is an image of a carbon nanotube itself. And And here we have a fluorescent tagged image of retina. So fluorescent is a fluorescent molecule. It's an organic dye, and that was used to get this image of the retina. So that is another nanoparticle that is used in imaging. And here we have a schematic of DNA origami. So basically you use synthetic DNA to which can be used because DNA has pairing, the base pairing. You can decide the structure of a molecule of assembled nanoparticle by changing the sequence of this DNA. And that's how people made this DNA origami, and then they made more complex structures. So this was just a 1D structure. Now this is a 3-dimensional structures which have been made with DNA origami. Okay, so now that we have a little bit of intro into the different particles itself, we can talk about the different applications of nanoparticles. So the first application that we're going to talk about is bioimaging. So we know that imaging has become so important in today's world, right? In terms of like MRI images or X-ray, and even if you just leave the biomedical field, if you just look at your phones or TV, you're always looking for higher resolution images everywhere. And that's relevant even in the medical field. People want better resolution images. They want to be able to see what is the issue with better clarity, and that demands the need for these nanoparticles, which can target specific, because of their size, because of how small they can target specific tissues, specific cells, and that will allow us to see those specific tissues and specific cells. So it really, what it's targeting in this application is the higher sensitivity that the medical field is demanding, and the nanoparticles seem to provide in terms of, because of their size and because of their targeting capabilities. So one of the places where they are used is the fluorescent imaging. As I showed in the figure in the last slide, fluorescent is an organic dye, which is a nanoparticle, and which has been used to image fluorescently tagged particles, cells. It's very highly used in imaging cells, and very, very specific. And there's been so much work that is done on it now that we are able to get really clear, high resolution images with fluorescent tagged molecules. Another huge area of research is using quantum dots, both for diagnostic purposes as well as therapeutic purposes. And for targeting purposes, people are really working on quantum dots. As I said, right, the stained glass example was how the silver and the gold were acting as quantum dots, and people didn't know that before, but that's the point. And they have a lot of potential to be used in targeting, in diagnostics, in looking at trying to, what I mean by diagnostics is that I want to know what a particular disease is or when it is occurring. For example, cancer. Cancer, as we know, is a progressive disease, right? And you want to know at an earlier stage whether you have it or not. And quantum dots can provide us a way, as a biomarker, as a way to detect these particles at an earlier stage and show that they are present in the body. So that's one way in which it is being used. Another area is the MRI contrast agents. So MRI contrast agents, MRI is basically magnetic resonance imaging, and it allows us to visualize the different tissues in the body. But one of the limitations is that similar type of tissues will look similar. So if you have a particular tumor, or if you want to find the location of the tumor, it might be difficult to do that. So the area of research surrounding MRI contrast agents is pretty extensive. And one of these nanoparticles also provide a segue into using them as MRI contrast agents. And one of the examples is the superparamagnetic iron oxide nanoparticles. The schematic here is an example of a group which used these paramagnetic nanoparticles. They coated it with heparin, and they characterized it first. So this is the point. So when we say that, I would like you to imagine experimentally or practically what this looks like, right? These particles are so small, you can't really see if something is coated on top of it or not. How does it look? Is it even there? And that's why these tools like transmission electron microscopy, atomic force microscopy, AFM, are very important tools, because they allow us to see, okay, this is the surface, this is the structure, and this AFM gives us the structure. Okay, it's spherical or it's not. What does it look like? It allows us to see these nanoparticles. So they first, any nanoparticle research will first try to characterize the particle, how it looks or whether it is present. And then we'll try to see whether there is some effect biologically happening. So the second figure here is just a schematic of how these iron oxide nanoparticles could be used for cellular imaging, right? So you have the nanoparticles and they are functionalized with PEG. Okay, so what happens here is that PEGs are basically used to coat these nanoparticles so that they are not taken up or eaten up by the macrophages or the cells in the body. Usually what happens is that whenever you put nanoparticles into the body, they will form, there will be a coating that will be formed on the surface of these nanoparticles. And it's called corona. I know. I know that might sound very scary right now, but that is what it is called, and that happened before COVID. So it's called as a corona. And to protect the formation of this coating is what you do is you put these PEG molecules, which are these big hydrophobic or hydrophilic molecules, which kind of repel these particles to come and coat on the nanoparticles, so they can be functionally still relevant. So that's why we have these functionalized nanoparticles. You put them into these cells and you label the cells, and then now you can image them and you can see these cells itself, the specific cells. Okay, so that is bioimaging. Another application of nanoparticles is as biosensors. So as I mentioned earlier about even like quantum dots, right? They use for imaging, but they could also be sensors. They could target specific cells, and they could act as sensors for specific targets, for specific biomarkers, and they can indicate whether a disease is present or not, or what is the amount of a particular molecule that is present or not in the body. And this can be done outside. So one way to do it is by making these arrays, the micro arrays or nano arrays, as we would within this scheme of this discussion, where you would have these particles, these nanoparticles on a surface, and because they are nano, you can imagine that the same surface will hold a lot more particles than if it was a micro or a macro scale particle, right? So just by the number itself, it allows for a higher probability of capturing a biomarker, and hence a higher probability of detecting a particular disease. In this example, though, it's a different way of using these nanoparticles, where in this paper what they did was they took carbon nanotubes. So the nanotubes, if you remember, the structure of it is kind of like a grill, right? It's interconnected. It allows for better absorption of other particles onto it. So it also helps to detect things better. So they try to detect these volatile organic compounds, 1,2,4-trimethylbenzene and decane, which are indicators of lung cancer. And using these functionalized carbon nanotubes, and they increase the concentration of the analytes here, the markers, and they saw that these nanotubes could detect it as it increased, at least for the TMB marker. The decane kind of saturated after a point, but the TMB increased with increasing concentration, which is a great thing because these volatile organic compounds are actually like, they are supposedly in the air, so as you breathe or the saliva, so it would be very clinically relevant to have something like this. So you can imagine that how one of the issues with nanoparticles or any biomedical research is taking it from lab from a benchtop to industrial or commercialized setting, and this paper kind of tried to attempt to bridge that gap here. Okay, so the next application we have is the antioxidant activity. So in the body, there are these free radicals that are generated. Okay, these free radicals can be from oxygen, it can be from nitrogen, it can be from polymers, it can be from phenyl groups, as is indicated by this figure here. So it can be from different parts, different molecules in the body. But these reactive species, they are also called as reactive oxygen species, which is derived from oxygen, and these can be very harmful to the body. So if it enters the cell, it will act on proteins and DNA, and there are proteins present on the cell membrane itself. So the presence of these molecules can cause damage to the cells. So it has been found that many nanoparticles can act as antioxidants and on different reactive species. And this figure kind of gives us an indication as to what kind of, so there are the metal nanoparticles affect the hydrogen-derived reactive species. There are some phenyl-coated nanoparticles which affect the phenyl reactive species. The organic nanoparticles affect the oxygen-derived. The redox-active polymers, they act on the polymer-based reactive species. So these different kinds of nanoparticles can act on these different reactive free radicals in the body. So the body originally does produce these antioxidants, but when there is injury or when there is some condition, when some disease condition, then this balance can be mismatched, and having the presence of these nanoparticles helps to kind of counteract the presence of these free radicals. Okay, another arena which might not come so easily to us is this application of antimicrobial agents, but it is still very relevant. So we do know, and this is something that we observe in our lives, right? Sometimes we develop, as human beings, we develop resistance to certain antibiotics. So we need to up the dosage. And we've seen cases of this even within COVID and what's happening in the world right now, where a certain amount of antibiotics is not enough. So you give so much to the body. And what happens is that when you give so much antibiotics or a large concentration, then the body itself develops resistance. And the pathogens, what's worse is the pathogens or the disease-causing agents, they themselves also develop resistance. Then what do? How do you treat a disease when the bacteria or the pathogen itself has already developed resistance? That means you have to start from scratch and create antibiotics again. So that's where nanoparticles have also shown some efficacy in helping to act as antimicrobial agents. So what happens is that they have these high surface to volume ratio. So imagine small, tiny spheres, right? If you imagine a bag and you have these spheres which are macro-sized in millimeters, and imagine if you have in nanometers. So what will happen is that you will be able to pack more of these spheres if they are nanometer size. There will be higher density packing, and just because of that that increases the probability by which these particles can interact with whatever it is in. If it's in a solution and there are cells, then it can interact more because of the high surface to volume ratio. And that's one of the major advantages of using nanoparticles for this particular application. And here in the summary, basically, it has shown us different ways by which this could be causing antimicrobial effects. So one of the ways, if we follow my mouse, you will see that the first way is, so the legend here is that the coated particles, the silver nanoparticles, the silver ions are the big silver spheres, and the stars are the reactive species. As I was talking about in the last slide, these are the reactive oxygen species which are harmful to the cell. So in this summary, they're talking about silver nanoparticles and which were delivered to the cell and have shown my antimicrobial properties. And when these nanoparticles can go and adhere to the cell membrane itself, they can interact with the cell membrane, and this can cause breakage of the membrane. You can imagine that, right? And then it will lead to release of these silver ions. So these silver ions can further cause problems. It will go inside the cell. It will disrupt the transcription of the DNA and cause problems there. And it can also create these reactive oxygen species, which is harmful to the cells. So this reactive oxygen species can then go into the Golgi apparatus or the mitochondria, which are all part of the cell and can disrupt the cell function. It can also impact signaling. So as these silver particles are in the solution inside the cell and outside, it can impact how the cells talk to each other, and that will also cause a lot of problems. So this would be disruptive to the pathogen or the bacterial cell, so and will help with antimicrobial effects. Okay, so then we come to this major field of application, which is cancer. And cancer has been a very dominating field in biomedical research itself because of its high rate of mortality, because of how progressive it is, and how, although there are treatments, the treatments cause more side effects and more toxic issues and problems, and people are constantly looking for ways to improve the treatment, improve the targeting, and to reduce the toxic side effects of using cancer drugs or chemotherapy. And as I said, the need is that the therapeutic pathways we need to find more better ways of treating cancer and to do more efficient targeting, so you're not killing both the tumor cells and the healthy cells, but only targeting the tumor cells. So some examples are carbon nanotubes have been used as anti-cancer drug carriers. So basically they're used for tumor targeting. So they carry these drugs, and because of the structure of carbon nanotubes, the drugs that adhere to the nanotubes better than other nanoparticles. So that's one example. Another example is manganese dioxide nanoparticles have been used for reducing the tumor hypoxia. So the tumor causes it causes an environment, and that environment is harmful to the cells, to the human body. And so this sort of nanoparticles helps to reduce that hypoxia, and it also helps to reduce the chemotherapy resistance. So if this part impacts the relapse and the chemo side effects that are associated with chemotherapy. And gold nanoparticles have been used for tumor targeting. As I mentioned earlier, it is important to target. So what is currently happening is that the drugs target both the tumor and the healthy cells, and because of that, it's very unhealthy for the body. And any means by which we can target the tumor cells around, and it's very hard. I'm saying it as if the medical field has not stepped up. It's very difficult because the tumor and the healthy cells are so interconnected. That is the reason why it is so difficult to parse out what exactly is a tumor cells. In a 3D environment, everything is just mixed together, and the tumor has its own blood vessels coming up, and it's very difficult to differentiate what is a tumor cell and a healthy cell. And that is the reason why these nanoparticles are also so important. This research is so important. So the summary figure here kind of gives us an indication of the different nanoparticles and the different areas. So the bioimaging. As I mentioned earlier, the SpIO2 particles, the mRNA contrast agents can also be used to target tumor cells alone. So that targeting can be specific for cancer itself. And that's what we see here. And there are drug delivery properties. And then targeting can be based on targeting, and then based on targeting, there's like a radiation-based killing of cells, the photothermal therapy, and radiosensitivity, etc. Okay, so having gone through these applications, I would now like to talk a bit about a biology-specific example, because so that you get a true understanding of what it means, what is the application of this nanotechnology in a biological example. So right now we're going to talk about bone tissue engineering. Just to give you an overview, bone tissue engineering basically involves three components, which is the scaffold, the cells, and the molecules that are delivered with it. So an example of bone tissue engineering is, for example, if you have a hairline fracture, it's possible that it's a small fracture, like a small break in the bone, right? It will heal over time. But if you have a major accident and the bone breaks, it's called as a long bone defect, then for the bone to heal, it will take a long time. And so what is required, what is done is that scaffolds are put in place there, like grafts, which are basically these structures which are filled with cells and nutrients and molecules, so that it will allow the body, it will provide the incentive for the body to grow new bone there. And it's a very active field of research. And in this example here, we are going to talk about bone tissue engineering in terms of scaffold, where nanoparticles can be used for the scaffold itself, for making those structures, or for molecules. So the first part is the scaffold itself. And one of the reasons why nanoparticles are very beneficial to use in this respect is because they kind of increase the porosity of the scaffold. So what does that mean? Porosity basically means the gaps in between the connections of the scaffold, and how far anything from outside can reach the inside of the scaffold. And you might think that that's a very small thing, but it's not. And because we have to make these structures which are compatible with the body, so they are not that, I mean, earlier they used to be made with metals and things like that, but we saw a lot of issues in the body in terms of biocompatibility. So now people have moved on to polymers and other more biocompatible materials. So their structural stability is not as strong as metals, right? You can imagine that. So having these porous structures is not very trivial. So that's the reason why nanoparticles are very beneficial in this respect. And so they can, and increased porosity is important because you need to supply nutrients from outside into the inside of the scaffold. That's where all the cells are. So nutrients need to be supplied completely throughout. So here, this paper is a very interesting paper. So it's actually a vascular graft. So it's not bone, but it's a aorta. It's trying to mimic the aorta, or an artery in the body, so which pumps blood into the heart. So what they did here is that they made nanofibers, and they controlled the orientation of the nanofibers, meaning that one, on the surface they had these sort of circumferential orientation. That means it's going in circles around, and along the inside of this graft they did it longitudinal. So what this actually did was it promoted a specific type of cell on the outside and a specific type of cell inside. So the inside of the cell, inside of the graft had more endothelial cells, so blood vessel cells, for because that's what the human body has, because it wants to because they wanted to mimic the real blood vessel. And the outside has smooth muscles because they pump. The muscle cells will pump the blood, pump the vessel. So that's the reason why they had these two different kind of orientations. And the beauty of this paper is that they were able to achieve that because of this technique called as electrospinning technique, which allows us to make these nanofibers in this particular orientation, to be able to control in which direction these fibers are formed. And that's it's really amazing that they were able to do that, and that they saw that cells preferred, that cells, a particular type of cells grew on particular orientation. That means they're that sensitive to mechanical forces. So that's what they showed here, that the stiffness and the orientation of this matrix, the nanofiber matrix, changes what type of cells grow. So basically how this happens is that the specific type of cells, the smooth cells or the endothelial cells, they grow from stem cells. These stem cells have the capability to differentiate into any kind of cell. And how they differentiate into different kinds of cells is based on external factors. And as we can see here, matrix stiffness is also one of those factors that definitely impacts that. So that is one example. The other example I want to talk about is the molecules itself, the drug delivery. So another place where nanoparticles seem to play a really crucial role is the delivery of drugs or growth factors or genetic materials into cells. And it's also particularly relevant for bone tissue engineering because there's a lot of research going on about growth factors and how the timing and the delivery, the localized delivery of these growth factors can impact or enhance bone growth and healing. So the spatial and temporal control of this delivery is quite important, and nanoparticles provide a way to do that in a controlled manner. And the control over the localized, the region of delivery, as well as the time, will allow a very stimulus-driven response, or it will allow it not to be recognized by macrophages in the body and not to be taken up at one time. You can imagine if there is like you just supply a huge quantity of something, and then if it's just immediately taken up or immediately destroyed, then there is no point. And especially for bone growth, like if you have a long bone defect, then it takes a long time to heal. So you need to provide these nutrients or molecules for that entire time. So that sustained delivery or controlled delivery is a very important aspect of nanotechnology or of bone tissue engineering. Okay, so this paper is also very interesting because it uses curcumin, or what we know as turmeric, and it studies how turmeric can help in bone growth. So what they did here is that, if you remember the liposomal structure, they tried to integrate this turmeric curcumin molecule into the liposomes, and they put it into a 3D printed scaffold, and then they looked at, because this was novel in the sense that this loading of this liposomal curcumin was novel, so they looked at the release of this curcumin, and they saw that I know you can't probably read it, but the slower graph here, the blue one, is the liposomes with the curcumin. So what they basically saw was that it could retain it. They could do a controlled delivery of this curcumin molecule when it was encapsulated in the liposomes. So they first did a characterization of the liposomes, and then they studied the cytotoxicity of a particular cell, the MG63. So what they were trying to talk about here is that curcumin has, surprising as it may be, but I mean, we know as Indians, I think, that turmeric has, very beneficial properties. But there is a growing area of research which is studying this as an actual drug to help with anti-inflammation, antimicrobial, antioxidant effects in the body. So there's very active area of research in many places. And so the way they try to study it in this is by the inhibition of a particular cell and the proliferation of a particular cell. So they want to inhibit the MG63, which inhibits the bone growth, and they want to make sure that the osteoblasts, which enhance bone growth, is growing. So that's what they found, and they found that the liposome encapsulated curcumin definitely helped with the process. This was a very interesting paper, which showed the impact of using nanoparticles for tissue engineering. Okay, so we talked so much about nanotechnology and its many applications. But it's also very important, as we talk about these applications and this exciting field, to understand its limitations. So on the lines of that is the feasibility question, how feasible it is to actually use these particles. At the level of research, at the level of laboratory, it looks amazing. You can visualize things with this, you can see things happening, it's moving, it's impacting the cells, that's great. But how can we take it from the laboratory to the real world, to the industry, to commercialization, to people? And so one of the emerging things in that is nanotoxicology, where people are now trying to study the toxicity associated with these nanoparticles. I know we talked about how we are using nanoparticles to combat the toxicity, but they also might have their own toxicity, and it is very important for us to understand the physical and the chemical properties of the nanomaterial, how it behaves outside the body, like in vitro, if it's just cells, how it behaves inside the body with the entire environment, the microenvironment of our bodies. And if you have done some sort of research, you would know that in vitro and in vivo behavior is completely different for many, many particles, and more so for nanomaterials, because the thing is, there's so much in biology that we don't understand. So it's important to study whether it's having any long-term side effects or what is the toxic effects of using these nanoparticles. And research has shown that it could have some toxicity due to penetration into these cell layers, because it could produce more of these free radicals, and we don't want that. And the dosage, the amount of nanoparticles you put might have an impact, and you might want to limit how much you put. So there was this growth factor called BMP2, which a lot of people add, which they used in a particular company, but they used huge quantities of it, and that caused bone growth everywhere, and they didn't know what to do about it. And it caused a lot of issues because of that. And that's the thing, especially with growth factors, it is very important, and which is one of the molecules, very important to control the dosage, how much you put in at a particular point in time for bone growth, for example. Another aspect is the commercialization. So making these nanoparticles, having worked with nanoparticles myself, I can say that they're very finicky. Making these particles and making sure that they are stable and is very difficult. And to make that at the laboratory level and to take it up to commercialization or industrialization level is much more difficult. So that's one of the limiting factors of nanotechnology right now, but a lot of people are working on it. And the efficacy and the fact that when you take it from lab or benchtop, you take it to an industrial setting, will it still have the same biological effect that you're observing in the lab? So that is something to also keep in mind. With that, I will end my talk. This is the references. Please let me know if you have any questions.
Thank you, ma'am, for taking us to the world of nanoparticles. The session was highly informative. There are a few questions from the audience. Let us start with the question and answer session.
Sure.
Participants kindly post your questions in the chat box. Ma'am, the first question is, what is the role of nanotechnology in antiviral therapy?
Sorry, I didn't hear what you said.
What is the role of nanotechnology in antiviral therapy?
Antiviral therapy, of course, I mean, it would be impactful even in that. I can't cite exact examples. If you have a more specific question, I could possibly answer that. But it is, I mean, nanotechnology is relevant even in antiviral therapies as well, because it could help in delivering certain viral strains. So DNA and genetic material is another component that can be delivered using these nanoparticles. So you can imagine that if you want to target specific strains, then you can do that using nanoparticles.
Thank you, ma'am. And the next question is, though nanoparticles are used in drug deliveries, in most of the cases, microparticles are used. Why?
So one of the important aspect is the size of the particles. So it's not, we can't say that microparticles is used or nanoparticles is used. Nanoparticles might not be used because they are difficult to form and assemble and things like that. And I wouldn't say that microparticles are used more. Maybe they are used, it will really depend on the application. So it depends on where you're putting these particles. And if they are in a place where micro-sized particles are going to be taken up quickly, then using micro-sized delivery particles will not help. You have to use something, you have to kind of fool the system, use something that is smaller so that it's not taken up by the body. So it really depends on where you are injecting.
Thank you. And the last question is, are there any specific health or other risks from nano product if given in vivo?
So, as I was saying in my last slide, right? It yes and no. There could be side effects, like if you are using ferric oxide particles or certain contrast agents. There could be side effects. That's why people want to use more like lipids or liposomes. That's why liposomes are so attractive, because they are basically made of these lipid molecules which are very similar to your cell membrane. The cell membrane has these lipid molecules, and these liposomes are made of similar structures. So it is easily digestible, or it's very biocompatible. So people prefer to use organic nanoparticles because it has less toxicity. But again, they come with their own issues of stability, because in the body, these nanoparticles, organic nanoparticles, are very fast. They're digested very fast. So if you're going to put it from outside, they will also be digested very fast. So if you want to have some sort of controlled delivery or something, it's not really useful to do that. So people are working on ways to still put the molecules, but have them in some sort of structure that they are not recognized by the body that easily. But yes, I think there is toxicity, but that's why people are moving into using more organic nanoparticles.
Thank you, ma'am. Now we are at the end of the question and answer session. That was a highly informative session, ma'am. On behalf of the management, IPAC, Department of Chemistry, Biochemistry and Physics, Animated Arts and Science College, we are grateful to Ms. Devika Krishnan for her excellent lecture on nanotechnology in biomedical research. Thank you, ma'am.
Thank you so
Nanotechnology
much.
in medicine. Thank you, ma'am. Nanotechnology in medicine is going to have a major impact on the survival of the human race in future. With this message, we are ending the first session of this international virtual conference on recent advances in biochemical and material sciences 2020. And a gentle reminder, we request the participants to fill the feedback form without fail and do rejoin for our second session at